All right, so we're going to keep on this journey with chapter 6. So, I always like to start off with right here is what is the goal of mitosis? Why do we have to do this? What is the point? Well, there's several reasons. If you think about right here, so I am sharing with you. Okay, sorry about that. You think about what your bodies consist of. It's mostly somatic cells. Make sure I had my view right on my side. The cells have to divide. Things happen. They get damaged. Think about when you get a sunburn. All right, so you went outside and sure you put on the sunscreen, but even when you put it on, you forget to reapply and you might get that sunburn. Well, what happened in that sunburn is those UV lights came and damaged your DNA, and your body recognized it's like, okay, these cells are damaged, so we need to commit them to undergo apoptosis, and we need to replace them with healthy cells. So that is one way right there. So replacement is a good one. Growth, organisms grow constantly throughout the time. So as we grow, it's not that their cell gets a lot bigger. Sometimes you have to get multiple cells. Technically, the size of a cell is kind of limited by its surface area. It can only get so big to where then it can effectively undergo metabolism. It has to do with surface area ratio and all this other mess. But, if an organism's growing, it has to have more cells. Another way is asexual reproduction. So this is how some organisms actually reproduce. Bacteria is a prime example. Now, if they're going to be using it for asexual reproduction, they're going to be clones. They're all going to be exactly the same because mitosis is just making the same exact copy. Now, remember to mention your body contains mostly somatic cells. You do have some adult stem cells. This is just a little diagram kind of walking you through your hematopoietic stem cell line, where I'm going to get all your different type of blood cells. This is how cells go through and make copies of themselves. It's the process of mitosis. Now I mentioned on the bacteria, or especially your prokaryotes, this is how they go through and do reproduction. Now remember, they lack a nucleus or other membrane-bound organelles, so they really don't have like a true cell cycle. Whenever it's time for them to divide, what they do is they take that circular chromosome, so they take their DNA and they duplicate it. And then they will copy both sides. Remember, they have a circular chromosome. And then eventually those two circles separate. And then the cell starts to elongate. Boom, boom, boom. And eventually what's going to happen is we're starting to get this little ring to form in between them called the septum. And eventually this is going to be split into two cells. This is called binary fission. So we're going binary, two cells to split. So we're going from one cell to two. How fast does this happen? Well, the life cycle of some bacteria is really short. I've seen it happen in about 24 minutes. It's really fascinating how fast some bacteria can divide. It depends on the type, but it can be quite rapid, which is quite fascinating when you think about it. But we want to focus on mitosis for this one. So we're talking about, our eukaryotic cells and how they go through the process of dividing. Now, following interphase, so if you remember the cell cycle, which I'll show you on the next slide, we're going to go into what's called the M phase, or mitosis. Now, we're going to divide it into 5 phases: prophase, prometaphase, metaphase, anaphase, or telophase. Now, with this, we're going to look for patterns in the cells. And the patterns are going to tell us what is going to be happening. So just by looking at this diagram right here, you can probably see some key things happening. It's like, oh, everything's lining up in the middle. Oh, the cell's looking like a peanut. We're going to look at these patterns, and then you should be able to, because I think this is on one of your assignments, to actually look at an image and say, okay, that's a cell in metaphase. That's interphase. That's a good anaphase right there. That's the goal of today, is to help you recognize and look at these different stages. Now I put here a note. Some books combine both prophase and prometaphase together. They just call it prophase in general. They'll call it early prophase, late prophase. I've seen it go both ways. I think for today, we're going to kind of keep them separate. Keeping the order in your mind, here's a little analogy. Please pitch mostly at the catcher. Whatever works for you. Everyone has their own little method. Another term that will show up is cytokinesis. This is where we actually split the cell from 1 to 2. And this is going to be associated with telophase most of the time. So just reminding ourselves about the good old cell cycle. So remember, in interphase most of the time, and then in phase. Now in interphase, we broke it up into 3 parts. We have that G1. This is where we pretty much grew a lot. Another key thing that I didn't mention before is centrioles. Now centrioles, they're going to be important because they're going to be our microtubule organizing centers. And what we did is we duplicated them in G1. So we had one set, we copied it into 2 sets in this phase right here. S phase is where we copied up all our DNA. We'll talk about that in the next unit. And then in G2 is where we go through and checked everything, duplicated all the organelles, and made sure everything is right. And now we're getting ready to go into this mitotic phase. And just to say next unit, it's a typo. I'll catch that. All right, so first one, prophase. Just call prophase on its own, or some folks will call it early prophase. Now what's going to happen is, if you remember when your cells are hanging out in interphase, I use that term plate of spaghetti. The chromosomes are going to be very decondensed normally. So you see my pointer circling on this cell right here. You see it very dark. If you see a very dark nucleus, it's a good indication that that cell is in interphase. Now, what's going to start to happen in early prophase is your chromatin is going to start to condense into the chromosomes. So we're going to start to wind up and start to make those little X's that you're used to seeing. So you're going to start to see, if you're looking at it, and you start to see some like organization. So the cell that I'm kind of hovering around right here, this is a great cell that's in prophase. So if you compare it to, you know, this good interphase right here, you see how it's really dark. You're starting to see some organized. You can actually start to see some of these chromosomes lining up. This is a good cell prophase just by looking at it. Now another thing that's going to happen is the nucleolus becomes smaller and disappears. And we also, if you know, that's in the very center of the nucleus. And what that's going to do is it's kind of be disassembled, because we don't need to make ribosomes right now. We are just committed to dividing. And we're going to just break it apart because we don't need it right now. We're going to start to form our mitotic spindles. So remember those centrioles? What they're going to do is they're going to start to move to opposite poles. Sorry. And as they start to move to opposite poles, we're going to start to make some microtubules that will grow between them. So prophase. Chromosomes condense. Nucleolus goes away. Start to establish the poles, and you start to see some early microtubules. Now, we don't see this on this microscope because we'd have to stain for it. We're just staining for DNA here. So, late prophase / prometaphase. At this point, chromosomes should be condensed. This is where we're going to start to break down the nuclear envelope. Now, the nuclear envelope is going to, we're just going to break it apart because we need to get the chromosomes out, because eventually we're going to move them towards opposite sides of the cell. So what we're doing is we're just going to break it down. We're going to reassemble it later. Now, at this point, the microtubules keep going, and we should have our centrioles have established our poles. So we'll have like a north and a south pole just by looking right here. Now, what happens is the microtubules are going to attach to our sisters. So we're going to have this protein complex called the kinetochore. And this is where the microtubules are going to kind of attach. It's going to be right by the centromere. Now this is going to be really important for the M-check phase. So in prometaphase, we're starting to make these connections. So these microtubules are starting to keep growing, and they're going to start to attach to these sisters. So at this point, envelope's going away. Chromosomes should be condensed. Poles are established, and we're starting to get our microtubules attached to them. So let's get to the next phase, which is metaphase. I always think of this as middle, middle. At this point, all the chromosomes line up what we call the metaphase plate. Your poles are clearly established, and all the chromosomes are now going to be attached to a microtubule on both sides. All right. Now this is where we're going to do that thing called the M checkpoint that I mentioned, the checkpoints throughout the cell cycle. What the checkpoint does is it does like a little tug test. So what's going to happen is microtubules are going to pull on both sides. And as long as the chromosomes don't fully go towards one side, then that passes the test. Now, if the chromosomes pull towards one side, it's indicating that a microtubule is not attached to the other side. So we do that to prevent non-disjunction from happening. So if I'm looking at a good example right here, this is a great one right here in the middle. I think this is one of the easiest ones to recognize. Everything's lined up in the middle at this metaphase checkpoint. When we do karyotypes, this is when we actually stain for cells to actually look at them. So metaphase, middle, everything lined up. Now anaphase, this is where we're going to happen. We're going to split the sisters apart and pull them towards opposite sides of the cells. So what happens is we kind of take out that staple, that centromere staple that was keeping these sisters together. We remove that. So now the sisters can be broken up. We can split them. And what happens is one copy goes towards this pole and the other one goes towards this pole. They are pulled by the microtubules. The microtubules start to push the cell too. It starts to elongate. So some microtubules are attached, others are not. And what we're doing is we're going to pull these chromosomes half towards this side, half towards this side. So here maybe one that's an early anaphase that I'm circling around. And here's a great one, probably in later anaphase. So you can see like half of it split over here, half of it split over here. So at this point, the chromosomes are completely separated. And then finally, telophase. All right, telophase. This is where it kind of starts to look like a peanut. There's several things that happen. So the spindle disassembles. We're going to break up this complex. So we're going to get rid of our microtubules, decondense that. At this point, everything's towards its opposite poles. The cell's elongated. What's going to happen are the chromosomes are going to start to decondense, get more towards that relaxed state. Nucleolus is going to reappear. We're going to reform a nuclear envelope around each of them. And then you will, at this point, the cell will have two nuclei. All right, so until it commits to cytokinesis, this cell has two nuclei at this point. If I'm pointing at an image, I would say the cell that I'm hovering on right here is a good cell in telophase. It's kind of one of the harder ones to find. So here's one nuclei, here's two. Cells a little bit longer. I would say this is a great example of a good telophase because the chromosomes are decondensing too. And cytokinesis. So cytokinesis is where we pretty much split the cell into two. So what's going to happen is we're going to form what's called a cleavage furrow in the cell. We're going to start to form this actin and myosin ring, and we're going to keep pinching and pinching and pulling to where eventually we go from one cell to two. And now these are two new identical daughter cells. Now with plant cells, it's a little bit different. So remember, they have the cell wall. So what's going to happen is we're going to put up a cell plate, which is made up of Golgi vesicles. It's going to show up. And then that's going to form, and eventually that will be converted into the cell wall. So this is a great review slide. It has a lot of the things going off, but I'll point out the key things. So in prophase, chromosomes condense. They start to see some space in between them. Microtubules will start to grow. We're going to establish our poles. Nucleolus goes down. We're going to start to break down that nuclear envelope too here. In prometaphase, the envelope should be completely broken down. Poles established. Microtubules are growing and starting to attach to the sisters. Metaphase, everyone lines up in the middle. We do the M checkpoint, tug, tug. Anaphase, split the sisters apart. Half go towards one pole, the other half go to this pole. Telophase, all right, cells elongated. nucleus repairs, nuclear envelope reforms, chromosomes decondense, we break apart the spindle complex, and the cell's elongated. We're at 2 nuclei at this point. And then cytokinesis is where we officially split it into 2 cells. So this is a cool little video. It kind of shows you several images. They're kind of sped up. You can see the chromosomes and see if you can identify the different stages. It's kind of cool. And you'll see one of them where there's a problem where, the M checkpoint, it seems like it's hanging out there for a while. It's because it was doing that tug test and it was seeing one of them wasn't lining up, or we had one chromosome just kind of floating around, not knowing where he was supposed to go. So it's a really cool video. We would have watched it in class. So I'm going to stop it right there. There's some videos on the next slide that kind of show you review.